ഇപ്പൊ തുടങ്ങിയിട്ടുണ്ട്.
Please
Sir, good morning, sir.
You can.
Sir, Niranjan sir, good morning, sir. Sir, good morning, sir.
Yeah, good morning. You can start, madam. Sorry, there is a technical issue. Some gap is there. I think load is more.
Good morning, sir. Namaste.
Sir, Mohana madam is online, sir. You can connect.
Am I audible to you, sir?
Yeah.
Okay, please.
Yeah. Yeah, but
ఒకసారి చూడండి చెక్ చేయండి రాలేదు వస్తుంది.
Hello.
Sir, good morning, sir. Is it clear?
Yeah, good morning. It is now audible. Okay, madam. You can start. Thank you.
Sir, thank you, sir. And today we have Dr. Mohana Madam online, and also today's inaugural chief guest, Dr. G. Narasimha, sir, are also joined in the meeting, sir. So with due permission, can we start the program?
Yeah,
sir?
thank you, madam. Yeah, please proceed.
A very good morning to the respected delegates. We, the team of Seven Hills College of Pharmacy and CSIR Institute of Minerals and Materials Technology, cordially invites you, all the delegates, for the 5 days virtual eFDP program on pharmaceutical innovations and protection strategies, scheduled in July 18th, that is today, to 22nd of 2023. And we have initiated this program in line to the National Intellectual Property Festival, which is commenced in order to promote the intellectual properties and also nurture the teachers for protection of intellectual property. At this juncture, I sincerely thank all the delegates and also organizations who extended your support and really bring up the life to the program. And it's a very great moment that we have received crossover more than 2,000 delegates from all across the country. And I sincerely applaud and gratitude from Seven Hills College of Pharmacy and Team CSIR for this great registrations and support from all the delegates. With this short note, I request everyone to join together for the prayer song. Thank you, sir. With this, we officially declare this virtual FTP program is open. And now I request and I welcome our beloved principal, sir, Dr. M. Niranjan Babu, sir, and the patron of this program, Dr. T. Pavan Kumar, sir, Senior Scientist, CSIR, IIMT, Bhubaneswar, to present the welcome address. Sir, thank you for joining in this meeting, and I request you to go for a welcome address on this occasion, sir. Thank you. Over to you, sir.
Om Namo Venkateshaya. Good morning Namaskar. I myself, Dr. Niranjan Babu, Principal, Seven Hills College of Pharmacy, Tirupati, Autonomous. On behalf of everybody, first of all, I want to thank Dr. T. Pavan Kumar Garu, Senior Scientist, CSIR, Institute of Minerals and Materials Technology, Bhopal, India, for materializing this event, 5-day virtual faculty development program on pharmaceutical innovations and protection strategies. Wholeheartedly, I am welcoming you, sir. Dr. Pavan Kumar, sir. I also welcome today's chief guest, Dr. G. Narasimha, sir, Department of Virology, Sri Venkateshwara University, Tirupati, Andhra Pradesh. You are welcome, sir. And we are conducting on behalf of CSIR to celebrate National Intellectual Property Festival. So this 5-day FDP will give you a beautiful outcome. For this, we have today resource persons, Dr. S. Mohan Lakshmi Garu, Director and Principal, Amity Institute of Pharmacy, Amity University, Gwalior, Madhya Pradesh, India. You are welcome, madam. One more resource person, Sri Amit Kumar Patil, Ahmedabad. Today they are going to join with us. Thank you. Once again, I want to welcome all the registered delegates, other resource persons for this 5-day FDP program. Have a nice day. Jai Hind. Over to Dr. Sucharita, madam, for further proceedings.
Thank you, sir. Thank you so much for briefing about this today's eFDP program on pharmaceutical innovations and protection strategies. Thank you so much once again, sir. And I sincerely welcome today's chief guest of the program, Dr. G. Narasimha Sir Garu from Sri Venkateshwara University. I welcome you, sir. And on this juncture, I request Professor K. Saravanukumar, sir, to brief the citations of today's chief guest, Dr. Narasimha sir. Sir, over to you.
So good morning to all. Myself, Professor K. Saravanukumar. It's my privilege to introduce today's chief guest, Narasimha sir. So Dr. Narasimha sir, working as Associate Professor in Virology Department at Sri Venkateshwara University, Tirupati. He had his doctorate in microbiology from Sri Krishna Devaraya University, Anantapur, India in 2003. He had 18 years of teaching and 25 years of research experience. His research area during his studies in applied microbiology included microbial enzymes, secondary metabolites, surfactant, characterization, and purification. After he joined the virology department at Sri Venkateshwara University in 2007, the growing applicability and problem-solving ability of nanotechnology tracks to his interest, and he combined his past experience with growing nanoscience and technology in finding solution to old problem in a novel way. He started guiding students in applied nanotechnology, biology, and green synthesis of metallic and nanometallic nanoparticles characterization and their application as potential antimicrobial, antiviral, and biocatalytic agents for modern medicines and agriculture. Dr. Narasimha sir published more than 150 papers in reputed national and international journals. He attended national and international conferences, symposia, and presented research paper, served as an expert member for scientific programs. He handled research projects and served as principal investigator for major research projects sponsored by the Department of Biotechnology, UGC, and Board of Nuclear Sciences. He published books and book chapter of international repute. He is serving as editorial advisory member for a good number of national and international peer-reviewed journals. He received national and international recognitions and awards like a highly cited research, Young Faculty Award, Distinguished Faculty Award, Research Fellowship Award from the various organizations. Thank you, sir. We are kindly invite for our program. Thank you, ma'am.
Thank you so much, sir. And I now request the today's chief guest of the program to give inaugural address on this occasion, sir.
Thank you. Thank you so much, madam. And thank you, sir,
Thank
for
you,
your nice introduction. So first of all, I highly thankful to the managing director and manager, and particularly respected principal, Seven Hills College of Pharmacy. And today keynote speaker, Dr. Pawan Kumar, senior scientist, CSIR IMMT, Bhopal. Dr. S. Mohana Lakshmi, director, principal, Amity University. And organizing committee, Professor K. Sarwan Kumar, Dr. P. Sucharita, Dr. B. Jyoti, and other faculty members, students, and participants of today's program. Good morning to all. So my hearty congratulations to management of Seven Hills College of Pharmacy and CSIR IMMT Bhopal, Bhubaneswar for the jointly organizing the 5-day faculty development program. I am very happy to attend this program. Today program related to the current topic like pharmaceutical innovations and protection strategies. So this area has the wide scope in the novel ideas and to make them towards the commercialization. So we have the several medicinal plants in nowadays also. We need the focus more research in this area also. Today, the pharmaceutical science is the wide and current novel field. There is a much scope for the further research also. The researcher should be the focused by the containing the recent domain like bioinformatics, nanotechnology, biomedical engineering, and artificial intelligence, so that the stability and design of the new compounds will be possible for the synthesis of the new drugs. Now the government of India also supporting the innovations, particularly in the domain of healthcare, diagnostics, and artificial intelligence. And even they are giving the funding programs like VIG, Big Grant, DBT, ICMR, DBT, etc. So hope the young minds and faculty will they can focus on the such type of innovations, and they can synthesize the novel drugs, and they can, the very important one is the patenting also very important. Once the innovation is over, the very important you must care about the patenting like IPR technology. Nowadays is IPR also very, very important. Even for the publication also very important. My suggestion to the students and young minds, do research in the innovative idea, particularly current era in the pharmaceutical science, and publish in the peer-reviewed journals like Scopus and Web of Science journals only. Because nowadays the UGC and DBT, they are looking only this indexed journals, not other journals. My suggestion to the young minds, please publish the journals in the peer-reviewed journals with a good impact factor. Then, and even patenting also very, very important to do the proper way. I hope the today eminent speaker, they will give the more information about the IPR and innovation technology. Hope the participants will gain the knowledge. Once again, I congratulate to the management of both managements for the grand, and wish for the grand success of this program. Thank you so much, sir, for giving this opportunity. Thank you so much, sir.
Thank you, sir. Thank you for gracing this occasion. And you rightly said the protection of the intellectual property rights is the true area where everyone want to focus, and that is the objective of this FDP2, to nurture all the faculty members in the way to protection all the intellectual ideas. Thank you so much for gracing these occasions.
Thank you.
Thank you so much. And on behalf of our principal, I request you to accept the honor of certificate for gracing these occasions, sir. And Principal, sir, I request you to present this certificate to the chief guest of this today's program.
Thank you, sir. Professor Narasimhan, sir. Please accept our.
Thank you, sir. Thank you so much, sir. Thank you so much, sir.
Yeah.
We need even collaborative
Thank
work
you, sir.
with our department also, sir. We have very good research lab, and our students also focusing on nanotechnology and novel compounds from the nanotechnology, like bioactive compounds, nanomaterials, for the particularly current emerging diseases, for the treatment of the emerging diseases also. Thank you, sir. Thank you so much.
Thank you, sir. We'll definitely approach you, and we'll have a much more collaborations with you, sir.
Thank you, sir. Thank you,
once
madam.
again, sir.
Thank you.
Thank you.
Thank you.
And moving ahead to the presentations of this today's program, this eFDP program comprises of totally 10 presentations. And to have a good kickstart to this program, we have our today eminent guest speaker who made significant contributions to the field of pharmacognosy. And thank you so much, ma'am. And it's our pleasure to have, and also you are going to inaugurate this eFDP with the first session. Thank you so much, ma'am, Mohan Lakshmi ma'am, for accepting. And I request Dr. B. Jyoti ma'am to introduce today's resource person, Dr. S. Mohan Lakshmi ma'am, to the audience. Kindly read out the citations, please, ma'am.
Thank you, Sucharita.
Thank you.
Good morning to all. Our respected principal, sir, today's chief guest and session speaker and teaching faculty and all delegates. Myself, Dr. B. Jyoti, professor and PharmD coordinator, Seven Hills College of Pharmacy. It's my honor to give brief introductory note of my mentor, advisor, and PhD supervisor, Dr. S. Mohan Lakshmi Madam Garu. Madam was born on 16th August 1977, completed her undergraduate course in pharmacy at the Tamil Nadu Dr. MGR Medical University, Chennai in 1999, and pursued her postgraduate degree and doctorate at Jadavpur University, Kolkata, which is the 6th university and 13th in overall category in NIRF ranking. She started her career as assistant professor in pharmacognosy at SRM University, main campus, Chennai, and worked in various positions such as assistant professor, associate professor, and principal in various pharmacy colleges. During her tenure, she is instrumental in starting PharmD many PG and PhD programs in the colleges. She is also the key person in driving the institution to 2F and 12B status of UGC and getting recognition for research center of the institution. She is a recipient of Career Award for Young Teachers from AICTE, Government of India, New Delhi, and the recipient of Fast Track Young Scientist Award from DST, Government of India, New Delhi. She is also the recipient of Guru Ratna Award from Government of Karnataka during Teachers' Day and recipient of Distinguished Professor Award from IPA Madhya Pradesh State Branch. She has visited Japan, Singapore, Bangkok, Turkey, Bangladesh to present her research papers. She has filed 5 patents, out of which 3 patents are granted already. She has guided 6 PhD scholars for their doctoral degree for Jadavpur University, Kolkata, JNTU, Hyderabad, and JNTU, Anantapur, and SRM University. At present, she is guiding 3 PhD students for their doctoral thesis. She authored a book in pharmaceutical engineering and also contributed many chapters for Bentham and Taylor Francis published books. She was nominated one among 50 women scientists across the country by the Department of Science and Technology, Government of India, to participate in the International Training Program on Leadership and Career Development for Women Scientists and Technologies. conducted at Indian Institute of Science Education and Research, Pune. She was also nominated by the Ministry of Human Resources and Development, Government of India, to participate in Women in Science Administration and Management at ISER, Pune. She was interviewed by the Deccan Herald newspaper to share her views about this program. She has participated in workshop cum training program on innovation, incubation, entrepreneurship conducted by Columbia Business School, Columbia University. She has successfully executed 2 research projects funded by AICTE. She published more than 50 research articles in indexed journals. Her index is 19 and I-10 index is 40. Dr. Lakshmi is the reviewer for journals published by Elsewhere and Taylor and Francis. She served as member Board of Studies of the JNTU University AP, served as member secretary for the IEEC for animal experiments and president for internal complaints committee, accuracy coordinator, a member governing body in various pharmacy institutions. She received an award for idea presentation on the topic creation of integrated platform for different sources of health data in idea sharing competition organized by IEEE. At present, Dr. S. Mohalashmi Madam is working as director and principal at Amity University, Amity Institute of Pharmacy, Gwalior, Madhya Pradesh. Thank you, Madam. Thank you one and all for this opportunity.
Thank you so much, Dr. Jyoti ma'am. And really, it's a blessing to every student to read the citations of the mentor like Mohan Lakshmi madam. And thank you, madam, once again. And madam, it's over to you. We can go for our first session of today's program.
Thank you. Thank you, Dr. Jyoti, for a very elaborate introduction. Am I audible to you, Dr. Sucharita? Am I visible and audible? Okay.
Yes, you are perfectly all right, ma'am.
Thank you. So, Honorable Chief Guest of this Faculty Development Program, Dr. Narasimha from Department of Virology, Sri Venkateshwara University, and Dr. Niranjan Babu Garu, the Principal of Seven Hills College of Pharmacy, and Mrs. Sumalata, the Chairperson of Seven Hills College of Pharmacy, and the patron of this FDP, Dr. Pavan Kumar, Senior Scientist of CSIR Institute of Materials and Minerals, and the convener of this Faculty Development Program, Dr. Saravana Kumar, Dr. Sucharita, Dr. Jyoti, and all the faculty members of Seven Hills College of Pharmacy and all the staffs, students, ladies and gentlemen. Good morning to all of you. And it is my pleasure to be connected with you virtually to deliver a talk on commercialization of pharmaceutical innovations. So Seven Hills College of Pharmacy is very special to me, very close to me, because I am seeing very closely this institution from the foundation level. And I am very happy and pleasure to join this institute virtually for this FDP program. My best wishes to the further growth of this institution. So once again, I thank all the organizers for giving me this opportunity. So let me start my presentation. May I share my screen?
Yes, ma'am.
So the topic for my today's presentation is commercialization of pharmaceutical innovation. So my talk is divided into 3 parts. So just before going to the pharmaceutical innovation exactly, I just wanted to inform the students about the general innovation. That will be the first part of my presentation. And then I will be talking about what is the difference between invention and innovations. And then I'll be talking about pharmaceutical innovations. And then finally, I'll be talking about the commercialization challenges of the pharmaceutical innovations and what are the advantages of this same. So first, let me discuss about what is invention and innovation. So people think both are the same. Usually they use these 2 terminologies as a synonym. So in the place of invention, they use innovation. In the place of innovation, they use invention and vice versa. but they both are, the both terminologies are, has a different meaning. They are not the same, they should not be used interchangeably. So how it is different? How the inventions and innovations are different? So to be an invention, first of all, invention is a creation of a product or introduction of a process for the first time. For the first time, if anything is invented, for the first time, if any process is discussed, then that is known as invention. But if this invention remains in the place where it is invented, if this invention is just like that existing, it does not have any meaning. So it should be used, its applications have to be experienced by the people and the society, then only that invention has got a meaning. So that is known as innovation. So when we improve the invention so that the existing product, the invented product has a significant contribution for its application, so that particular part, that particular process is known as innovation. It could be used, its applications have to be, something is wrong in the audio?
Dear participants, please mute your mics, please. Thank you. Yes, ma'am.
May I continue?
Yes, ma'am, you can. Thank you.
So for further explanation to understand more about what is invention and innovation and what is the difference, for the sake of the students, I will give few more examples so that they will understand better. So in the picture, you can see a wheel-like structure which is made up of stone, and there is a vehicle which is having this wheel fixed in that vehicle. So the first time wheel-like structure is first time discovery or first identification is known as invention. But if this structure remains in that place, it does not have any application. So if it is fixed with any vehicle so that that vehicle will be made movable, then that process is known as innovation. So you can understand how the invention of wheels have made a revolutionary in the history of transport. So without wheels, we can't just imagine our life. From the trucks to the turbines, today wheel has become important part of our life. So here, this stone-like structure is an invention, whereas this vehicle which is fixed with these wheels are the innovation. So this is the second example, a general example, which is not related to pharmaceutical. I just wish to give this for another better understanding. So this is the picture which depicts a person who involved in the printing process called Gutenberg, which taken in 1455 year. This picture shows that he prints the first paper, the first page of Bible in a paper, and they are very excited to see that because he is the person who first transported the ink onto the paper in the form of letters. So he printed 42 lines in one page, and this is the Bible which is first printed, which is kept in the left side of this image. So they are very excited to see that letters have been translated into a paper, and this was sold for 30 florins. 30 florins is equal to 3 months of salary of a person who is working in the printing press. So that was the amount equivalent. So they printed 42 lines on single page. So this is a well-known example for inventions and innovations. So in 1876, what happened with Alexander Graham Bell, who has tried to make his voice to listen after a long distance by another person is an invention. Whereas afterwards, what happened with the cell phones, today Apple iPhone and all that is all are innovations. So this is another invention, that is the fire. This fire was known 1.5 million years ago that it is invented by just, crashing two rocks, how the fires are produced. And today, you know how much the fire has revolutionized in many, many applications. So this is an invention which is a magnifying glass, which happened first in 2068. So people who invented this magnifying glass in 1268 is not the invention, it is only an innovation. So what is the invention? Invention is by the Egyptians who used some form of crystals to see and improve their visions to see the letters in the bigger size, which happened in 700 BC. So that is an invention. Afterwards, what happened? Afterwards, the makeup of magnifying glass in 1268, and then now today all optical glasses and the glasses which are used to magnify the images and all that are all innovations out of this invention which happened in 700 BC. So this is a very well-known story of the Wright brothers invented the aeroplane. So there is a short story behind this particular, it is not actually called invention. I will just tell you why it is so. So Wright brothers were known to invent an aeroplane first time, which flied for 12 seconds in 120, for about 120 feet higher. But what has excited them or what has inspired them to do this particular experiment? But this is not the first human flight. So what is the invention behind this? What is the first invention behind this? The first invention behind this are two printers, two people who involved in printing press are the inventors of this first invention, who made this first invention of this aeroplane. So what they have done, in a short, it is a big story, but in a short, I will try to explain in a few lines. So these people have, while printing, they observed in their room, when they exposed a particular plastic bags or any other bags which is made up of any fabrics, when they exposed to the hot air, this paper will rise. So what they understood, when a material is heated, then it rises, it becomes lighter than the air, and then it rises. So with this, they made a big air balloon, and then they started flying it. And this experiment have fascinated them that why can't a human also fly into the sky? So first they demonstrated this experiment to the king of France and also other dignitaries in front of them on 1783. So they have just hanged in a bag, one sheep, one duck, and one rooster. And then they allowed that hot air balloon to fly. So this is the picture which shows that hot air balloon is flying, where that sheep, duck, and the rooster are suspended below that balloon. And all the dignitaries and so many people, lakhs and lakhs of people are witnessing this demonstration of this particular experiment. So why they have decided the sheep, duck, and the rooster to be suspended? Why they have selected this particular 3 species, which also has a scientific backup? The king actually suggested them to use prisoners for this experiment, but instead that inventors have decided to use these 3 species. A sheep is having the same biological factors like a human, and the duck and the rooster. The duck is a bird which will not be harmed in the higher altitude, and which is used as the control here. The duck is used as a control here, and the sheep is used here as an alternative to human, and the rooster is used here again as a second control, but the control which will not fly higher, which will fly only to a short distance. So with these 3 species, the balloon flied successfully for a few minutes, and then it landed unharmed. Even the animal bird species was also not harmed, and the balloon was also not harmed. So this is the main inspiration and invention for the Wright brothers to invent the aeroplane. So again, I will give one more terminology for the difference between invention and innovation. Invention requires knowledge, competence, and financial aid, because in empty hands, we can't demonstrate anything. Whereas the process of innovation involves identifying the market needs, trends, developing competencies, and finding financial support. So this is the difference when we try to invent or innovate things. A spreadsheet software is an invention. Using a spreadsheet software A business application will be made and profits are increased are known as innovation. So a quality for a patent for invention. So all inventions are not eligible for patenting. So a little bit, patent is not my today's topic. Just to connect this invention with patent, I am just putting this line, this sentence. So every inventions cannot be patented. An invention which have to be patented must pass a test of originality, and it should be sufficiently different from the previous invention. And most inventions are minor improvements on the existing invention which do not qualify for patents. If you make only a small changes of existing inventions, then you can't proceed for patenting. Only a small percent of patented inventions, even though if you patent, still every patent will not have economic value. We can't say I have patented, now I can make a lot of money, now I can commercialize it, and then I can sell it, I can make a big profit, etc. A patented invention, all patented invention will not have economic value. So why it is so, we will see in the further slides. Now I am going to touch the pharmaceutical innovations. In this topic also, I am going to give you a few examples of the olden ancient innovations and the latest innovations for a better understanding, because we should not think that the invention, the terminologies like inventions, innovations, critical thinking, design thinking, or entrepreneurship, startup, these kind of terminologies are not new. They are not recent or modern. Since the ancient time, innovation exists, invention exists, and they all served as a foundation for today's modern innovations and inventions. Let me move on with pharmaceutical innovations. So this is a very well-known example for each and every one. You may be knowing this. You might have heard about the Alexander Fleming's invention of the penicillin. So what happened with Alexander Fleming? He was testing in the laboratory with the Staphylococcus bacteria. So he put the bacteria, he just inoculated it in his petri dish. This is the picture showing that working in his laboratory. So he left it and he went on for a short vacation. So after he returned back to his laboratory to see the petri dishes condition, and he found lot of growth on the petri dishes, which is not familiar to him, which is something different, which looked like a mold. And he observed this mold, whichever this bacteria which he has inoculated have shown no growth wherever this mold are present. The mold is found everywhere, but wherever this bacteria is there, that bacteria have shown no growth with this presence of this mold. So it means he understood this mold has some killing effect of this bacteria, or this mold has some effect of slowing down the growth of this bacteria. It didn't allow the bacteria to grow further. So with this invention, he started identifying what is this mold and what is present in this mold which kills this bacteria, or which slowed the growth of the bacteria. So with the help of a group of people, he tried to identify this. So this is in 1935 taken picture. He presented a petri dish with penicillium mold to his colleague. And this is a group of people who helped him in identifying or separating this penicillin, because penicillin, even now it is known for his allergy producing effect. And when it is first time used in the form of crude, it produced a lot of allergic reactions, and people started purifying it. And the quantity of penicillin that was obtained from the mold is very, 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 very less quantity. They don't have place to grow this mold. They used in the hospital whatever they had. This is the picture which shows the materials which even the bed pan which is used in the hospitals were used to grow the mold, the fungus, and then they used all these materials which were available in the laboratory and in the hospital where they have used for the growth of this mold. So they employed a few ladies who are known as penicillin girls, who helped in purifying this penicillin process. because in tons and tons of penicillin, they got very few milligrams of tons and tons of broth, they got very few milligrams of penicillin. So that was a big challenge to, get the penicillin in the pure form. So this is the crude penicillin, which is obtained from the urine of person who was given penicillin. So because they found it very difficult in isolating penicillin from the broth, so they collected the urine of a person who was given penicillin for some treatment, and that urine were used to recover these penicillin, and this is what shows that. So, this penicillin have served as an invention for a breakthrough in the field of antibiotics. Today, we have number of antibiotics for using to cure various various infectious diseases. So this is another success story in the field of vaccinology. The vaccines were made. The first time vaccine was made by Jenner in 1749, 17th May. He experimented on the first vaccine. He given cowpox vaccine to people to protect them from smallpox. Initially, it had a lot of challenges. So Edward Jenner is a person who demonstrated first time on vaccines with the cowpox vaccine from a dairy maid. He injected it to humans. So he found very few mortality rate of people who had the smallpox. So he published, even though he invented this in 1749, he did this experiment on human in 1796. And in 1798, he published this research in publications in research articles. And in 1823, he passed away. But still, even at that time, people are not so positive about this vaccination procedures. But in later 1853, vaccination with cowpox was made compulsory in the British and World Health Organization announced the smallpox has been eradicated in 1980. So without his success story, this would have not been possible, that eradication of the smallpox. So today, how much of vaccines we have. We eradicated polio. We found vaccines for Ebola. We are now experiment on monkeypox. We found for influenza, Zika virus, COVID-19, and even for the future, the vaccinology department is ready to find vaccines. So this is the success story of discovery of X-rays. So Röntgen is a person who discovered this X-rays. So I just kept the picture of Röntgen, and this is the X-ray which they are trying on the person on 1895 November 8 to find the bullets which is lodged in his chest, which is found by the X-rays. And then this is the picture which shows her wife's hand with a ring in that by means of X-ray. So this is the story of anesthesia. So I just wanted to show you a small video before the invention of anesthesia, how the surgeries used to be made. So the person will be fully conscious. The persons will be knowing, they will be experiencing the pain that during the surgery procedure. So what happens with the surgery? This is a small few minutes, I think 2 minutes of video, how the surgeries are made without anesthesia.
Before the introduction of ether anesthesiology, surgery was just brutal, terrifying. The patient would already be inebriated, alcohol and other kinds of substances. It was incredibly bloody and gruesome. The intervention of left.
Understanding of how blood clots, an embryonic understanding of anatomy and what blood vessels, what structure.
So the person who underwent this surgery have given his experience in a journal in the form of article, because he had an amputation only with drinking some alcohol. Only he was fed with alcohol. We have seen in 1847 what he wrote in the journal. The horror of great darkness and the sense of desertion by God and man, bordering close on despair, which swept through my mind and overwhelmed my heart. During the operation, my senses were preternaturally acute. I still recall with unwelcome vividness the spreading out of instruments, the twisting of the tourniquet, the first incision, the fingering of the sawed bones, the sponge pressed on the flap, and tying the blood vessels, the stitching of the skin, the bloody dismembered limb lying on the floor. So can we imagine this situation now? So without anesthesia, the amputation of legs, hands have been done, and any kind of surgeries were done without anesthesia in the olden days. So after this in 1846, first time the ether anesthesia have been discovered by Henry Jacob Bigelow. He is a surgeon, and this experiment was published in Boston Medical and Surgical Journal, which is also known as the New England Journal of Medicine in 1846. So what is the invention behind this surgical procedure? So it is the story of Dr. Martin, who was a dentist, who tried to spray some vapor of some gaseous substances on the tooth of the patients, and then he extracted the tooth, so without any pain. So this was observed by the other surgeons, especially Dr. Warren, who have tried maybe the same materials we can use to do surgical procedures. So this is the first surgery made with ether anesthesia. You can see in this picture, which is a picture taken in the medical college, all are medical students, and the Morton is a dentist who is spraying the ether from his hand. You can see the small vessel he is having and spraying, and Dr. Warren is removing a tumor from the neck of the patient. So throughout the surgical procedure, it is explained that the pain is not experienced by the patient, and the patient have just only shown the murmur sound. So this is the anesthetic history, and with this anesthesia today, you know how surgeries have been revolutionized, and so many surgeries are being performed for the outcome of the patient. So this is another success story of invention. He is Werner Forsman. At this time of the experiment, he is 25-year-old medical intern who is a surgeon. So he is very fascinated about surgeries related to heart. At that time, there was no surgeries. At that time, no one touched the heart. So what he did, he himself inserted a catheter in his vein, and he pushed that catheter into his vein further until it touches the heart, only with some local anesthesia. So after he inserted that catheter in his own vein for 65 centimeter length, then he rushed to the radiology department and he taken a radiography. This is the picture of that radiography. You can see the catheter which enters into its vein until the heart. So this procedure is known as heart self-catheterization procedure, which happened in 1929. So you can see that also proof in the online, it is available. And the X-ray department have taken this X-ray, and his name is Forsman, 25-year-old surgical intern. And this is published in a Springer Link Publisher journal, in a German journal in 1929. This is the publication about the self-catheterization of the heart. So with this invention, later on, open heart surgeries were performed. And today the angioplasty, the ballooning techniques and all is due to this experiment. So today we have, with all these advent of this surgery procedures, today we have minimization of invasiveness in the surgery. Laparoscopy, thoracoscopy have been, used where the incisions are only half meter to half centimeter. And what is going to be the future? By the introduction of endoscopy and percutaneous techniques, now the incisions are not there, only puncture wounds are there. So because of this, the surgeries can be done only with local anesthesia, and they can be conscious, and the time and cost will be minimized, and there will be early recovery and early return to the work. So what is going to be the future? So how the present generation, the youngsters, can innovate from here. So what are the success stories I have shared for the surgery? What are the inspirations that I have shared with you? Now, what would be your innovation is robotic surgeries, devices which has only millimeters in size, like mini Medi boots, like a small Medi boots with a very few millimeter in size, which will go through the nose or go through the eyes, lungs to deliver drugs, collect tissue samples, install medical devices inside the body. This is going to be the future, and what the present generation can work on this for the future medicine and future surgery. So as very light overview, I will talk about the repurposing, because it is a very important topic, at the same time very well-known topic, especially after the COVID. So repurposing is identifying the new use of the existing material. I will just share you only one story of HIV. With that, I think you will be able to understand clearly what is meant by repurposing, instead of giving definitions, instead of giving things about repurposing. Only one example will be enough to understand this process. So on June 5th, 1981, this is the picture which shows a medical report. The medical report is called MMWR. MMWR is nothing but Morbidity and Mortality Weekly Report, which used to be published in every week, which lands on every desk of the clinicians. Similarly, like any other week, on June 5th also, it is landed on the desks of every clinicians, and the clinicians have read the report. What the report says, which is also mentioned here, 5 young men, all are homosexuals. 5 young men, all are active homosexuals, who were presented with a rare pneumonia called pneumocystis carni pneumonia. They are from Los Angeles, California. 5 homosexuals, they have pneumocystis pneumonia. So there is no connection between their sexual habit and the pneumocystis pneumonia, because these kind of pneumocystis pneumonia are seen only in those patients who have a compromised immune system. It does not have any connection with the sexual practice of the homosexuals. So what happens in the next report, followed by this, when the clinicians read this report, they were not sure what is going on with these homosexuals, and so why the homosexuals are getting this kind of disease, which does not have any connection with them. So this is the second report. Now the report says there are many homosexuals, nearly 26 in New York, California, the age between 26 to 51. They are presented not only with pneumocystis pneumonia, also with a rare cancer known as Kaposi sarcoma. So this report was also read by all the clinicians of the Los Angeles, where this reports used to be published. And they were not sure even now why this homosexuals are getting these kind of bizarre diseases. So on December 10th, few weeks later, on December 10th, 1981, the New England Journal of Medicine published a series of reports only on homosexual men. You can see this, there is pneumocystis pneumonia, mucosal candidiasis, and there is an outbreak in this disease, and there is perianal ulcerative herpes simplex. So many infectious diseases have come. At this point of time, the clinicians understood that there is an outbreak of new disease. something is wrong, which is there is an outbreak of a new disease, and this disease is not any existing disease, it is a new one. So at this time, you know very well about this person, Anthony S. Fauci, who spearheaded the COVID-19 vaccines, who is 83-year-old infectious disease specialist who works in US NIH, National Institute of Health. So he mentioned in his commentary about this infection. He is the one who read about this weekly reports and thought about it much, and then discussed about his peers. And he's mentioned in his commentary, it is not a publication, it is a commentary in the Annals of Internal Medicine, which is published in 1982, 1 year later of this invention. Because we do not know the cause of the syndrome, any assumption that the syndrome will remain restricted to a particular segment of our society is truly an assumption without scientific basis. So what he's trying to say with this commentary is, because we are seeing this infection in a particular community, that is the gay, that is the homosexuals, we can't assume that this infection will come only for the homosexuals, which does not have any proof, which does not have any scientific basis at this point of time. So please be careful. It may spread to the wider demography. This is what the Anthony Fauci, the scientist, wanted to tell to the public. But whatever he said also does not have any data, but it happened. What happened? The infection from the homosexuals spread to the heterosexual partners. It spread to people who received blood transfusion. It spread to people who are pregnant and they passed their infection to the newborns. The female sexual partners transferred the infection to their male partners, etc. And then it is named as AIDS immune deficiency syndrome, acquired immune deficiency syndrome, etc. Within a year and so, it becomes a pandemic. So we all know at that time, before the COVID, this is the biggest pandemic that happened, which has severely burdened the health institutions, severely burdened the health community. So people don't know at this point of time, the scientists don't know what is responsible for this infection. It is a new infection. Within 6 months, the patients are dying in the hospital and in the clinics. And there is a social stigma also attached to this infection. Those who got HIV were driven away from their house and they were not being taken care by even their own relatives. So research also parallelly started immediately. People started taking the blood and started testing, but still the life expectancy was only 6 months. The people are dying within 6 months of the initial observation. So this is the first publication, which is in the journal Science in 1984, 4th May, detection, isolation, and continuous production of cytopathic retrovirus from AIDS patient. So he is the one who identified and connected the infection with a virus. It is the virus which is a causative organism for this new infection. So the research started immediately. Now the repurposing comes into picture. So usually when there is an outbreak of disease, people and scientists, doctors, they don't have anything for new infection to treat. They don't have any medicines or molecules immediately to treat the infection. What they have is the existing medicines which were used for other purposes were tried on this virus. So there are so many anti-cancer drugs, there are so many antiviral drugs, so many antibacterials, antifungals, all anti-infectious drugs were used to be experimented on the new organisms which is responsible for the outbreak of the new infection. So they started experimenting similarly with this virus also. They isolated the virus, they have cultured the virus in the laboratories in mass quantities, and they have used all bunch of molecules that were already available on the shelf for other reasons. And with this, only one molecule, that is the zidovudin, AZT, there at that time, now it is zidovudin, which completely blocked the growth of this virus. So this is how the repurposing has become an invention of a new indication. And today this repurposing has a very big value, which is also an innovation, we can say, has a very big value because it reduces the time and cost for the discovery of new drugs. With HIV, afterwards, this virus started producing resistance to this. This microorganisms will do what they want, what they have to do. They produce resistance to the molecules, and then we have to produce new molecules. So new molecules of HIV were, synthesized by delineating the structure of the virus. The new molecules were integrase inhibitors, protease inhibitors, reverse transcriptase inhibitors, and the fusion entry inhibitors. So all these things will have a specific function, like it blocks the protein synthesis, it blocks the processing of assembly, it blocks the entry, it blocks the fusion, it blocks the integration, etc. So that today we have different classes of drug which has a striking and dramatic effect in the treatment of HIV. So this is all because of a basis of an innovation which happened due to repurposing. So that's why I wanted to say here, repurposing is also an innovation which one can concentrate on. So now I will give a few examples of innovations which is happening recently. So India is a diabetic capital, and many are suffering with the diabetes. There are a huge section of people who don't even know that they are living with the diabetes, and those who are suffering with the micro and macrovascular complications, mortality is more, etc. Even though there is more drugs and many different devices were invented and innovated, still this particular thing has not been, there is no permanent solution for this. So artificial pancreas system is the latest one in this. So this has a system with 3 components. One is the monitor, which continuously monitors the glucose level in our blood, and an insulin pump, which is an artificial beta cells, which will release the insulin whenever there is a elevated glucose level. And then this can be stored in a smartphone. So there are 3 components, and this can be installed permanently into your body for a certain period of time. So this is how it can be, fixed into your body, in the hip, in the hand, etc. And it monitors continuously the glucose level in your blood. And then whenever there is elevated glucose, based on the dose, based on the quantity of the glucose, the insulin will be released from the artificial beta cells, which is present in this system. So this has been approved by FDA also. This is known as artificial pancreas. Now people are innovating on it to make it more portable, to make it more smaller in size, to make it minimum invasive, etc. So this is a breakthrough in the field of medicine, which is known as flying pharmacy, which is made by drone. The UPS is known as unmanned drone. It is flight forward program, which happened in North Carolina. So what the first time was done with the help of approval from Federal Aviation Administration is the drones supplies bloods, urine, tissue samples, transfusible blood, and other medical essential supplies like life-saving drugs, etc. Even today, the vaccines are also added into it to a places which is inaccessible to terrain, during any climatic conditions, during rain, during severe climatic conditions, etc. So this deliveries were made in very few seconds. I will just show a small video here. So this is showing the challenges that we face when life-saving things were taken through on road in vehicle. If the roads are not good, So this is delivered in 15 minutes. So many companies are now involved in robotic supply of medicines, especially in Indian government are now pioneering with collaboration with foreign countries. So recently, Indian government have purchased many robotic things for the border security, for supplies to the hill during the, during the flood, etc., and also for life-threatening diseases. AIIMS Rishikesh is the first to take 20 kilograms of anti-tuberculosis drugs through drone for an urgent supply. This is another video which Zipline released recently. This drones, how these drones, the new model of drones.
Over the last 7 years, we expanded dramatically and we now operate in 8 countries. We serve 3,400 hospitals and health facilities globally, and we'll be serving 10,000 by the end of this year. Places for the 2 and 50 miles away, Platform 1 is going to be the right way to do that. Platform 2 is going to be the right way to make deliveries in denser settings.
This is how the drones are getting charged in the docking centers.
And then it's as simple as wrapping up the package like they would normally for any delivery, whether that's lab medical tests, a home delivery of prescription, a food delivery to someone's home, loading it into the droid through the loading portal, closing it, scanning both, and then sending it off to be delivered to a customer. We can typically guarantee, it's 7 times faster than an instant delivery via traditional service you might buy from today, whether you're ordering food or ordering something online. Much faster, it's zero emission, and it's way more reliable. The reality is, like, the vast majority of packages, on the order of 80–85% of packages being home.
So this is the picture what Indian government have done for robotic deliveries, what we are doing during robotic deliveries, and ICMR is now collaborating with some foreign countries to bring a iDrone project where they will supply the medical essentials through drones between hospitals and for life-saving treatments and life-saving supplies. Now the last topic, which is I will be covering it in a very few minutes only. So commercialization of innovation. So commercialization, what is meant by commercialization? So now there is an invention and there is an innovation on that invention. And now there is an innovation which have to be used by the people. So it cannot be used at a free of cost. So people want to make money out of this. That is known as commercialization in its simple terminology. If at the same time, the person who is inventing or innovating don't want others to use his innovation, then he has to protect his invention by means of intellectual property. This is a simple terminology I have explained to you. So any innovation, if it has a profit in terms of money, then it is known as commercialization. If the return is in the form of economic return, if the return is in terms of monetary benefit, then it is known as commercialization. So an invention and innovation does not always have economic return. Suppose if a person don't want to take an economic return, suppose don't want to patent, let the wider public use it, then that will not have a commercialization significance. So a person who is inventing or innovating, if the impact is on environment, on technology, on political, health, societal, it may have any kind of impact. A innovation may have environmental impact. A innovation may have some change in the climate, may change in the water quality, may improve agriculture, etc. Yet an invention may have some improvement in the existing technology. An innovation or invention may have any improvement in the existing political situations or existing existing health issues, society issues. They are all not known as commercialization. If the impact is only for environment, improvement of technology, improving political situations, health, society, then it is not commercialization. If the innovation has only economical significance, if the innovation was used to make money, then it is known as commercialization. So this commercialization is transfer of knowledge from one party to another, transfer of knowledge to another party and gaining some money or gaining some economic is known as commercialization. Now coming to the pharmaceutical innovations commercialization aspect. So pharmaceutical industry has a different scenario compared to the other industry. So what is the different scenario here? If any inventions or innovations happens, they must get a special approval, special permissions from a regulatory body. It is not like any other innovations. It should, when this innovations in the pharmaceutical field to come to a market, it need to get special approvals from the regulatory bodies like FDA, etc. And pharmaceutical industries innovations related to pharmaceutical industries should have a scientific backup. They should have some scientific breakthrough, some scientific discoveries must be the basis of these innovations. And these innovations cannot be accepted without any experimental and clinical data. They must always have a scientific proof and scientific background for these kind of new products and processes of the pharmaceutical industry. So if a person wanted to protect it from others to exploit, then he need to protect them from there by applying to the intellectual property, applying through intellectual property. So this intellectual property are the other speakers may detailedly explain about this. So the pharmaceutical innovations commercialization should always have a science or technology driven background, and they should be always backed up with the science and technology. So usually the pharmaceutical innovations are discovery of new products than the designs. Designs comes in other field, whereas in pharmaceuticals, more value comes from the new products rather than the design. We all know that very well. So I think with this, I can close my session. Thank you all of you for patient listening. If you have any doubt, you can interact with me. Over to Dr. Sucharita.
Thank you so much, ma'am.
Thank you, ma'am. It's a very good topics actually.
Thank
Exciting
you,
and interesting.
Thank you.
Thank you, ma'am. You have taken the presentation very logically, stating with what is the differentiation of invention, innovation, and gone for what are the principles of innovation, and finally you landed up into the commercialization aspects for the pharmaceutical products and also the other innovations. Thank you so much, ma'am. It was a beautiful lecture once again, and requesting the delegates, if you have any queries, you can unmute your mic and you can clarify with madam, or you can post your queries in the chat box. And before that, madam, we have a patron of this program, Dr. Pawan Kumar sir. Sir is a senior scientist of CSIR IMT. I welcome you, sir, for today's program, and thank you so much for being a great collaboration with Seven Hills College of Pharmacy, and thank you so much for giving.
Hello, ma'am.
Yes, sir.
The system, drone system, is been implemented in which state actually?
It is actually first introduced in US only, and it is very nicely used now in African countries, and India is also now part of it. Even today, 2 days back also, they have made a collaboration with for purchase of 32 drones for medical supplies. That is, going on very, very well.
Thank you. Thank you.
Dr. Pawan Kumar, sir, welcome you, sir. And here we have a professor of this first session, Dr. Mohan Lakshmi Madam, and Madam is a principal of Amity University, Amity Institute of Pharmacy, and happy to connect with you, sir.
Yes, yes. Can you hear me?
Yes, perfect, sir.
Yes, yes. So thank you very much, and I appreciate the efforts of Seven Hills College of Pharmacy in organizing these kind of programs. And today being the very first day, and the very first session by Dr. Mohan Lakshmi was really awesome. I was like, of course, I could not join through Zoom platform all throughout, but I was following it through YouTube live streaming. So really the kind of examples and the kind of videos and the kind of connectivity that she has taken in her presentation is really appreciable. And I hope all the participants would have got a decent understanding on the topic. And Amity University, yes, we do work in collaboration with Amity University from CSR IMT Bhubaneswar also. And Madam Dr. Mohan Lakshmi, really, we are very thankful to you as part of organizing team. I also convey my appreciations to you once again. Thank you.
Thank you.
Thank you, sir. It's all your efforts that we are here today. Thank you so much for accepting for a great collaboration.
Wish you all the very best.
Madam, can you please stop sharing the screen?
Is that okay now?
Yes, yes, ma'am. Perfect. And I request you to kindly accept our honor for the efforts you have made today, ma'am. And I request
Thank
Dr.
you
T.
so
Pawan
much.
Kumar, sir, to kindly present his felicitations to Dr. Mohan Lakshmi, madam, sir.
Yes, yes. So should I read it?
No, sir. No,
Ah.
you can present it. You can present
Okay,
it.
okay,
Thank you so much, sir. Thank you for this felicitation. Thank
Yes,
you.
Madam Dr. Mohan Lakshmi, this is a presentation of appreciation certificate to you on behalf of the organizing team of this program. So we are very much thankful to you. And
Thank
we
you,
look
sir. It's my
forward for
pleasure.
continued association. Thank you.
Definitely, definitely it will be there, sir. Thank you.
Thank you so much, madam, for accepting this and making our first session of this virtual faculty development program an informative one to all the expectations of the faculty members. And I hope all the other remaining 9 sessions will be so much fruitful to all the enthusiastic faculty members. Thank you so much from Seven Hills College of Pharmacy, the
Thank you,
management
Dr.
and the
Suchita.
principal.
Thank you. It's my
Thank you so
pleasure.
much, ma'am. Thank you.
Madam, thank you so much, madam.
Thank you, Jyoti. Thank you.
And delegates, here is the end of the first session of the first day of this eFDP program. And the feedback link will be shared after the completion of the second session by 3 o'clock in the today. And I request all the delegates to join for the second session of this today exactly by 2 p.m. with the same link what we have given in the morning. Thank you so much for joining and connect with us for this FDP program. And I request all the delegates to comply with the rules and regulations of this FDP. That is 80% of attendance is mandatory to avail the e-certificates. So kindly attend the program and make this objective a fruitful one. Thank you so much to all the delegates. Namaskar.